Hello everyone, welcome back to my channel Microbiology Simplified. In this video PYQ series, I'll be discussing about ZN stain, that is Ziehl Neelsen stain. It is an important stain and it is done in routine process. So we'll see the MCQ asked in this topic and we'll discuss all the points relevant to this topic. Okay, so let's start with the video. So the MCQ asked, a 30-year-old man working as laborer presented fever and hemoptysis. Microscopic examination of a stained smear of a sputum revealed following organism as shown in the image, and what should be the sequence of reagent used for staining the smear. So what could be the organism? If you see the color of the organism in the stained smear, it is of pink or red color in structure. Okay, so this pink or red structure is which organism? Is Mycobacterium tuberculosis. Okay, and why I am saying this? Because this Mycobacterium tuberculosis is a acid fast bacilli. So what is acid fast and what is the stain which is used for this acid fast and what is the sequence of this stain, I will tell you in this today's discussion. Okay, so let's start the discussion. So ZN stain or Ziehl Neelsen stain, this is what type of stain? It is a differential stain, and what it will differentiate? It will differentiate into acid fast and non-acid fast. And what is that acid fast and non-acid fast? So if we will see ZN stain, so whatever the primary stain is used, if we will try to decolorize that primary stain by using a acid, it will not get decolorized if it is acid fast. So that organism will called as acid fast. And if it is getting decolorized by the decolorizer, that is the strong acid used, then it is called as non-acid fast. So acid fast will not get decolorized by the acid used as decolorizer, whereas non-acid fast will get decolorized by the acid used as decolorizer. And the Ziehl Neelsen stain is actually a modification of Ehrlich's original method, in which aniline gentian violet is used as a primary stain and strong nitric acid is used as decolorizer. Okay, so we'll see the reagent which is used in Ziehl Neelsen stain or ZN stain in short. It includes primary stain, decolorizer and a counter stain. Okay, the primary stain used is carbulfixin, decolorizer used is 20% sulfuric acid, and counter stain is used is methylene blue. So this is the most common reagent which is used for ZN stain: carbulfixin, 20 to 25% sulfuric acid and methylene blue. So we will see the reagents one by one. So the primary stain, if you see, it is carbulfixin, and the concentration of carbulfixin which is used for ZN stain is 1%. Remember this. 1% concentration of carbulfixin is used for ZN stain. And it is composed of what? It is composed of basic fuction, in which it is phenol, which is a carbolic acid, is added to it. Phenol, which is a carbolic acid, is added to basic fuction. Okay, and this phenol acts as mordant, which will helps in firm binding of dye to the cell. Okay, so this is the primary stain, carbulfixin. Okay, now the decolorizer. So decolorizer can be primary decolorizer or secondary decolorizer. Primary decolorizer, which is commonly used, is 20% sulfuric acid. As per National Tuberculosis Elimination Program, this sulfuric acid concentration which is used for staining ZN stain smear is 25%. Okay, and okay, and this secondary decolorizer used is 95% ethanol. So why there is a need of secondary decolorizer when a primary decolorizer which has strong acid is used? So the secondary decolorizer is used to identify the alcohol fast. So what is there is a need to identify the alcohol fast. So as we know, this tubercle bacilli is acid fast, but along with acid fast it is also alcohol fast. So it is both alcohol fast and acid fast. However, the commensal inhabitants are only acid fast but not alcohol fast. So that is why if we will do the secondary decolorization by using alcohol solution, we can be able to find the both alcohol fast and acid fast, which is only tubercle bacilli. However, the non-pathogenic commensal inhabitants such as smegma bacillus, which are only acid fast but not alcohol fast. Okay, so it will increase the specificity of the stain. So to avoid this, what we can do? We can use an acid alcohol decolorizer, which is composed of 3% hydrochloric acid in 95% alcohol. Okay, So we can use acid alcohol instead of using a primary decolorizer and a secondary decolorizer separately. Okay. And the counter stain which can be used, it can be methylene blue, which is most commonly Loeffler's methylene blue, and malachite green. Okay, so these are the counter stain which can be used, either methylene blue or malachite green. Okay, coming to the principle, if you see the structure of cell wall of mycobacteria, you can see the mycolic acid, which is a long chain fatty acid present on the outer layer of the cell wall. Okay, this mycolic acid is resistant to aqueous base stain such as Gram stain. Okay, and when the primary stain, the carbon friction, is applied in presence of heat and the phenol which act as a mordant in this carbon friction, it will stain the cell red. Okay, and once the cell is stained, it resists, this mycolic acid resists the decolorization and the cell remains red. And when the secondary stain, the counter stain methylene blue, is applied, it will only stain the non-acid fast structure blue, whereas the acid fast structure will remain red. So this is the principle of Ziehl stain. So if you see the steps, so in the first, cells are present on the slide, and when the primary stain is applied along with heat, for around 5 minutes, it will stain the cell red in color. Okay, and when the decolorizer such as acid alcohol is applied, the acid fast positive bacilli will remain red. However, the acid fast negative bacilli will become colorless. And when the counter stain methylene blue is applied, this acid fast positive bacilli will remain red. However, the non-acid fast bacilli will stain blue in color. So how it will look like? It will look like as shown in this image. Okay, this red color organism are acid fast bacilli, whereas this blue color organisms are non-acid fast. Okay, these are non-acid fast. Okay, and also if you can see the other structures, these other structures taken up the counter stain color, that is blue. If instead of methylene blue, if we have used the malachite green, the other structure will take, the non-acid fast structure will take up the green color. Okay, but not the red. If it is red, it is acid fast. Okay, and what are the various other methods which can be used to stain the mycobacterium? So in case of Ziehl stain, what we have done, we have used the heat to increase the permeation of crystal carbon friction into the cell. So that is why it is called as hot method. Okay, so Ziehl stain is hot method. Remember this. Which one is hot method? Ziehl stain, because we are using heat for the better permeation of the stain into the cell. So there is another cold method in which we are not using heat. What we are doing, we are increasing the concentration of carbon friction. Okay, so we are using more concentrated carbon friction, so we don't need heat. And what is this method is known as? This method is known as Kinyon method. Okay, so this is Kinyon method. Kinyon method is cold method. K for cold, K for Kinyon. Okay, so Kinyon method is cold method, Ziehl stain method is hot method. Okay, remember this. There is another method apart from this hot and cold method known as Truant method, and this Truant method is basically auramine rhodamine fluorochrome stain. And what we use in this auramine rhodamine fluorochrome stain, very simple. Primary stain is auramine O and rhodamine stain. Decolorizer which is used is 1% acid alcohol, and counter stain is potassium permanganate solution. Okay, so these are the reagent used for auramine rhodamine fluorochrome. And how it will appear? It will appear like this. So the tubercle bacilli will appear like yellow luminous rod. You can see yellow luminous rod in dark field. In dark field, when observed under fluorescent microscope. Okay, yellow luminous rod, tubercle bacilli, whereas non-acid fast bacilli will appear dark in color. There is no fluorescence. Okay, So this is two-hand method, Oramin Rodamin fluorochrome stain method. Okay. Now for Zn stain for weakly acid fast organism, Mycobacterium lepri is a weak acid fast organism compared to tubercle bacilli, and for that the declarizer used is 5% sulfuric acid. Nocardia, which is an actinomycetes, it is also weakly acid fast, and Coccidian oocyst, the oocyst of Coccidian parasite, those are also weakly acid fast. Together for them we can use either 1% sulfuric acid or 1% acid alcohol, and this method of staining this Nocardia and Coccidian oocyst is known as modified Quinion's method. Okay, modified Quinion's method is used for Nocardia and Coccidian oocyst. Okay. So what are the list of acid fast microorganisms? All Mycobacterium are acid fast. Remember this. Second, actinomycetes, which includes Nocardia, Rhodococcus, Sucamorella and Gordonia. Remember, if you have gone through this actinomycosis video, I have told you Actinomyces israeli is, although it is actinomycetes, but it is non-acid fast. Only Nocardia, Rhodococcus are acid fast. Okay. Legionella migdadi, this is also acid fast. Oocyst of coccidia such as Cryptosporidium, Cyclospora and Cystoisospora are also acid fast microorganism. Okay, so which of the following gram positive organism shows following appearance on Zn staining? So gram positive showing following appearance means, if you see, these are the red color filamentous organism. So red color filamentous, that means acid fast filamentous organism, also must be gram positive filamentous organism is which one? Actinomyces. No, actinomyces is non-acid fast. Mycobacterium lepri and tubercle bacilli are non-gram positive and also not appear in filamentous form. So which one is the correct answer? Nocardia. Which of the following are acid fast organism? If you remember the list which I have told you, Nocardia, yes. Cryptosporidium, yes. Cystoisospora, yes. Actinomyces, no. Mycobacterium tuberculosis, yes. So what is the correct option? Option C, 1, 2, 3, 5. Okay. So I hope this topic is complete and you will be able to answer all the questions related to this topic. So just go through this topic and make your own notes and you will be able to answer all the questions from this topic. Okay. All the best. Take care.